Hello, and welcome to this second video on Atom Ruins Everything, Lab Mice Edition. This video follows on from the previous to explain why an animal model is needed and cannot be replaced by other equipment, tools, and reagents. The crux of Atom Ruins Everything was that animal models are not a good indicator of human results in clinical trials or other research activities.
Out of all clinical trials based on mouse models, the results don't apply to humans over 80% of the time.
This really only serves as an example of how little they understand of clinical trials, animal models, and research in general. To briefly summarize clinical trials, they follow three primary phases of testing. The first involves healthy volunteers who receive gradually larger doses until the expected dose is reached without significant adverse events. The next is a small-scale pilot project with patients, and this proves the theory and expected results. It also identifies the side effects in patients. The next and final stage is composed of gradually larger trials with more patients involved. There is technically a final follow-up stage that is strictly not part of the clinical trial. This is closer to product safety monitoring and records all significant adverse reactions from the product on the market. In order to get to clinical trials, the initial research must be completed in animal models according to most regulatory procedures. This is called preclinical investigation. The simplest reason for this is the ability to demonstrate the same basic principles in a research animal as it would be in a human. Testing of drugs in non-human subjects gathers efficacy, toxicity, and pharmacokinetic information. Pharmacokinetic information is also often abbreviated as PK. Animal models are needed because any disease is complicated. Atom Ruins Everything thinks that these complications can be distilled down to a single overly simplified process using cells or other reagents. Cells are complex. There is no argument on that point. Humans are made of cells, and as a result, the complexity cannot be seen as a simple multiplication of the number of cells, but as an exponential factor of complications. For example, cancer cells are presented as the only factor that influences cancer itself. However, this is not true. Cancer is regulated by genes, the immune system, and more. To understand why cells and other means alone are not enough, an example is best. As an example of how complex this can get, a well-funded, well-understood, relatively simple area of research is blood pressure. It has one effect and only has one variation in the diagnostic criteria. Unlike cancer, which is almost as varied as the human body has tissues. Blood pressure is a somewhat more simple disease, with the symptoms of systolic blood pressure above 140 mm mercury and diastolic over 90 mm mercury. Further, an increase in blood pressure increases blood pressure regulating hormones. This leads to secondary diseases such as atherosclerosis and other kinds of cardiovascular disease. Blood pressure is regulated by the contraction of smooth muscle cells in the arteries and by nerves which increase the pressure. This is meant to be reduced by the kidneys, and they take out salts. These remove a volume of blood which follows on from the salt. High blood pressure is achieved through mechanisms in the vascular system, kidneys, liver, heart, lungs, and brain. There are interactions at the tissue level, where contraction of arteries leads to a local contraction of more smooth muscle tissue. This is treated by the use of a range of medication. The oldest of these are diuretics. These increase the amount of fluid removed by the kidneys, and that decreases the blood volume. There are more drugs which target heart rate, hormones, and salt levels. This is the summarized understanding of human hypertension. The hormones and cell signals released as a result of high blood pressure include adrenaline, noradrenaline, aldosterone, cortisol, angiotensin, and some more again. These target smooth and cardiac muscles, exo and endocrine tissues, hormone-secreting and hormone-regulating tissues, and more. What you see here is a limited map of some of the complicated pathways involved. These increase or decrease the salt concentrations, the volume of blood, they increase or decrease heart rate, smooth muscle contraction is hyperpolarized. There is very little that can be done to reduce this to a simple formula involving only one particular avenue of investigation. Here are some experiments that attempted exactly that. Taking arterial muscle cells, they were purified and refined to only those that could be grown in cell culture, and where smooth muscle cells remained. These were cultured. These were then exposed to gradients of noradrenaline and adrenaline in a time series. This led to a dose-response curve in genes which were turned off and on by exposure. The genes being examined were those associated with high blood pressure and inflammation. Only a limited range of markers could be used, because not all of these genes are normally active, let alone in a pathological state in smooth muscle cells. The smooth muscles are a tiny fraction of how the larger mechanism behind hypertension works. HPLC was used to identify and quantify how much of the hormones and neurotransmitters are present in this media before and after the fact. This identified 8 possible candidates that had varying effects and mechanisms, the most pronounced of which was noradrenaline. This media was taken from the cell flasks as only a simple sample of the possible candidates, and it is as close as you can get using a single tissue sample in cell culture. This cell culture was able to provide a depth of information in a reasonably affordable manner in a relatively large volume. It was limited by specificity to only one cell culture type. This means that data only applies to what happens to one cell type in isolation. Unfortunately, the human body and other organisms are like an ecosystem. Everything interacts to some extent. Look at this picture of a blood vessel. It is not just smooth muscles, but several layers of differing content. There are nerves, muscles, support tissue, and connective tissue. This provides a path to many other organs and influencing factors. These factors affect not just the tissues in the immediate proximity, but possibly all over the body. Compare this with the parallel of an animal model. The above was in conjunction with an animal model of atherosclerosis using the C57BL6. This had a lipid disorder gene that had been inserted by the supplier and drug-induced hypertension. The closest possible model to a human with hypertension is this mouse at the moment. It expressed the same problems, genes, and proteins overall. The same pathologies and atherosclerotic plaques and organ damage was seen. A very close match to humans, and a solid background against which the human clinical data could be compared. These mice were kept in cages of 5 and take up an area of about half a square meter. They had blood pressure, heart rate, and more monitored. The animals were culled and tissue collected, including the liver, kidneys, heart, brain, blood vessels, and serum. This covers most of the tissues involved with hypertension. The animals also give an idea of toxicity in both the whole organism and specific tissues from the drugs they were given. These models can range from brine shrimp to pigs and everything in between. Testing of drugs in non-human subjects is meant to gather this information on efficacy, toxicity, and pharmacokinetic information, and therefore guide future research in non-human species and then later human research. While preclinical research answers basic questions about a drug's safety, it can't substitute studies of the ways in which drugs will interact within the human body. When you consider the human body can be in a whole range of states that just do not exist in the animal world, whether this is the very young, the very old, the pregnant, the adolescents, more again. Therefore, before pharmaceutical companies start clinical trials on a drug, they conduct extensive clinical and preclinical studies, and these involve in vitro and in vivo cultures. These use a wide range of doses, and these obtain preliminary efficacy, toxicity, and pharmacokinetic information, and these tests assist the pharmaceutical companies to decide whether or not that they should follow this drug further. Therefore, you can get a basic set of biological data from mice along with other preclinical organisms. This whole organism, tissue-specific toxicity, genomic, proteomic, and pharmacokinetic data is important going into a clinical study. A lot of this information will keep a small lab busy for months depending on the numbers involved, especially so in a research institution like a university or other small lab, and more so for R&D departments which are often run lean. Before you can even get to an animal model though, there are barriers that have to be overcome. And there are several steps that have to be completed for those barriers to be beaten. The first of these are the 3 R's. And the first of the 3 R's are replacement. This includes cell culture, like that described in an attempt to circumvent the need for animals earlier. Cell culture is used to assess several areas. The first is cell effect. In the case of cancer cells, do they die when exposed to a certain chemical? Is there a functional difference? Is there a morphological or cell structure difference? Things like deposits building up inside the cell, as is claimed in the case of talcum powder. This gives an idea of how the animals might respond and what can be done to streamline it, and how to replace them if possible. The second is general tissue type effect. An example of this is paracetamol and hepatocytes. It was found that hepatocytes specifically die when exposed to excess paracetamol. This is important when you understand that this may have an effect on other studies or other particular experiments you want to run. Damaging the liver can lead to brain damage, kidney function loss, heart disease, all manner of things that you don't know about until you run that initial experiment. The next is toxicity assays. Using hepatocytes again, it can be estimated how much will cause irreparable damage and therefore be toxic to humans based on initial research using cells. Next are the genomic changes. It was found that the expression of cytochromes change in hepatocytes when they're exposed to this, and that's because they have to express more of the cytochrome to accommodate the paracetamol, as it is the mechanism that removes the toxicity from the liver cell. And the final area are proteomic changes. What proteins are translated or blocked in the toxic state, as opposed to others. Or if you are treating a cancer cell line, what is being secreted that prevents the immune response in this cancer cell line? The next R is reduction. The data found previously should give an idea of how to reduce the number of animals needed and how it can be done. The last R is refinement. Refinement is experimental design for animals that allow a researcher to get as much data as possible with the least number of animals and the least amount of pain, suffering, and term of their life. As you can see, this does not give basic biological data though. It does not give you anything tissue specific or toxicity specific in a complex organism. An example of this is thalidomide in humans. It was eventually identified that mice having been given this drug actually gave birth to defective offspring. However, that wasn't being looked at when it was first studied, and as a consequence was only realized when humans had issues with this drug being used. The latter two of these are necessary for a clinical trial. Tissue-specific information and toxicity information. This will tell you how much to give and whether or not there are any immediate contraindications and who would be a suitable candidate. The first is needed to demonstrate a cause and effect relationship. We are not allowed to give people random things wondering if it will make them feel better. You are required to have some idea of what effect a drug will have. Therefore, giving someone a drug on the premise that it will reduce blood pressure and it has no effect whatsoever is simply not okay, and it demonstrates no cause and effect relationship. However, an example of this can be found in Viagra. It did reduce blood pressure, but it was very ineffective in that role. However, its side effect is a little too well known now. Another option includes computer modeling. These can evaluate promising avenues of investigation before anyone need ever enter a lab. There are many databases of interaction between genes and proteins. There are databases being built on chemical and drug interactions, and there are more databases again on interactions between drugs and genes. And then there are drugs, genes, and tissue databases. In the future, these may be an option for research instead. However, it requires a far better understanding of how the world works than science currently has possession of. In the lead up to these experiments and investigation, the literature is scoured for leads, or the researcher builds on previous work. In order to ensure that their work fits into the larger body of existing publications, they must be able to structure it in such a way and justify it to an ethics committee, a funding body, and generally to peers and supervisors. These often include panels of experts who know what they're looking at and can judge something on its merits with that expertise. Further to that, should someone choose to publish in this field, it undergoes a process of peer review. Generally speaking, these people are well known, well versed, and well educated, and quite high profile within their area of expertise, and they will evaluate the content of an article and then decide whether or not it needs refinement, alterations, or corrections. This area then becomes something that has been evaluated by multiple people, looking at the publication, looking at the particular article and saying, this makes sense, this fits into what's already there. And it is the very first step in creating a drug lead or development. Thank you for watching this video. Please post any comments, questions or suggestions you may have below.